So this quarter, we have covered these steps used in this popular software package for single cell data analysis called SIRAT. So I want to say that in this quarter, I changed the course to discuss the computational statistical methods in this one analysis pipeline in the same context. What I hope is that I hope we use the same data from end to end that may help us understand those concepts in a simpler, unified setting. That's my hope. But I want to say or clarify that many of these steps are not unique to single cell data analysis. So don't think that, oh, I only learned single cell data analysis in this quarter. No. So I think single cell is just the setting. It's just the application. But many analysis or methods are used widely. So just to review what we have covered. So first of all, data normalization, which we spent quite some time talking about in the first 2 weeks, is something used widely in bioinformatics. And the purpose of normalizing the data is twofold. First, you want to make your numbers comparable. So in a single cell setting, we want to make the numbers for one gene, we want to make its values comparable across cells, so that when the value is higher in one cell, you can confidently say this gene is more highly expressed in this cell than other cells. So normalization is for comparability. And also, the special thing about single cell data is that whether you believe that the cells should have the same library size or not, because that's a different perspective. And in SURAT, the log CP10K method by default will normalize each cell to have a total count or cell library size as 10K, 10,000. And the assumption underlying that is you want to look at genes relative expression levels within cells instead of absolute expression levels. Because if you change this total count to 10K, you have removed the absolute value differences already, since some cells might be bigger than other cells, and that bigger should be reflected in the bigger library size. That's what we expect. So therefore, I would say how to properly normalize single cell data remains a controversial topic, and there are different aspects. So like one aspect we ask you to try in your homework is whether you have variance stabilizes given the mean. That is, you try to make the variance unrelated to the mean. And that is a property of Gaussian distribution. So for Gaussian distribution, you have two parameters. One is the mean parameter, one is the variance parameter, and they can be unrelated to each other. So therefore, this normalization to make the data Gaussian-like is to hope that we can apply some techniques designed for Gaussian data, Gaussian distributed data, to our normalized data. That's the hope. So therefore, normalization plays a crucial role. And there are also some other normalization ways so that you don't make data Gaussian-like, but you just want to remove the cell library size effect on your gene expression value, but you may still retain your gene expression as count. So therefore, there's no, I would say, unique way to normalize your data. It really depends on what you want. Like, do you want to make data Gaussian-like? You already want to keep counts? Or do you think data has the self library size effect that you want to remove or you don't want to remove? So all these decisions will need your justification. And I think the best justification should come from science, should come from your understanding of data generation and why you think one is more reasonable than the other. So this is actually quite challenging. It requires expertise, your understanding of the data, and your understanding of statistics. So I just want to say that default is not necessarily the best. If you have better justification, you can go away from default. But I think for most users, they use default because default has been tested empirically and found to be a robust option. Okay, so that's about normalization. One thing I forgot to say before we do the normalization is that we filtered out some bad quality cells and unexpressed genes. Okay, we did both gene and cell filtering. So for the cell filtering, the hope is that what we retain in our data is likely single cells. And we hope to throw away empty droplets that doesn't contain cells, and we hope to throw away doublets or multiplets that contain more than one cell. That's for data quality consideration, using our knowledge about the data. And for gene filtering, we want to make sure that the genes we keep are informative. So therefore, if some genes are consistently unexpressed across all cells, they are not likely informative, then we want to throw them away. Okay, so we have filtering, normalization, then scaling. So scaling is basically a simple step. You centered every gene's normalized levels to be mean 0, and you standardize it by dividing the standard deviation of the gene. So the gene has mean 0, standard deviation 1. Why is that? The reason is for the next step, when we do PCA, principal component analysis, we know PCA will preserve the total variance of all genes. That's the goal of PCA. And if we don't scale the data so that some genes have larger variance than other genes, then those large variance genes will play a bigger role in determining PCA. And we don't want that. We hope this PCA step is kind of fair for all genes. So that's why we do scaling before you do PCA. This is a standard practice in people's use of PCA. If you don't want the large variance variable to dominate the PCA, otherwise the first PC will be very similar to the large variance variables, so that they will be highly correlated. The first PC, if you have single gene that has the largest variance, then the first PC will be dominated by that gene, and that might not be the thing you want. So that's why you standardize every gene to have the same variance or standard deviation as 1. But in some applications, if you believe that the variances are naturally informative and you want to preserve the observed variance, then you can skip the scaling. So again, that step needs justification. Okay, so for the linear dimensional reduction part, we also cover several methods, including PCA, right? And for single cell data, we have another variant called GLM PCA. GLM PCA is different from PCA because we don't assume the data to be Gaussian. Since PCA, the nature of it is to decompose the variance, I should say, or to reduce dimension, but preserve the variance. So it naturally assumes that variance is the meaningful quantity, and it has nothing to do with the mean because we can shift the mean anywhere, but preserve the same variance. And since we do the centering step here, the mean is already removed. We use the centering to remove the mean, then we preserve the variance. But so it has this assumption that variance is informative. But we know that variance is informative for Gaussian distribution, but it may not be so informative if the variance depends on the mean. And that's when we have count data. So for both Poisson distribution and negative binomial distribution, which we briefly covered them in the lectures, their variance depend on the mean. The larger the mean, the larger the variance. So therefore, if you use count data, you do PCA, then it's not proper because when you do the centering and scaling, the centering alone is not an effective way for removing the mean effect because the mean effect still carried in the variance. So it makes the situation complicated. Therefore, GLM PCA uses basically the GLM, generalized linear model framework, to kind of do PCA within. So PCA is done on its transform parameters. It's in the inner layer, while it models counts directly. So it doesn't require this normalization, which is conceptually advantageous. But it is slower than just PCA. Since PCA has a close, how to say, eigen decomposition algorithm, which is not iterative. So you don't have to go through a complicated optimization to do PCA. But for GLM PCA, the algorithm optimization is the bottleneck. So therefore, there's a trade-off between this, I would say, elegance, conceptual simplicity, and also the computational time. And also, moreover, whether it is reasonable to assume that in the generalized linear model GLM part, is it reasonable to assume the data are Poisson distributed, negative binomial distributed, or zero inflated negative binomial distributed? That's another question you need to decide, because the model has to be reasonable for GLM PCA to work well. So you know that Poisson is a special case of negative binomial, because negative binomial has one additional parameter called dispersion. While if you go to zero inflated negative binomial, you have another zero inflation parameter. However, the good news is that for single cell data with the UMIs, unique molecular identifier, this step that removes PCR amplification bias, using UMIs, there's abundant empirical evidence showing that negative binomial is just good enough. You don't have to go to zero inflated negative binomial. I think that's good news. But the choice between Poisson and negative binomial, I think for both data, depending on how you, depending on how you model your data. For example, you may see the data is more like Poisson if you are more conditioning. That means you are looking at a very pure cell subpopulation. Then every gene count may be Poisson. But if you have a cell type that can be not so homogeneous within, like the cell type may have cells at different cell cycle stages, for example, then negative binomial may better describe the data. So that is another thing to consider. So anyway, like cell type, cell state can be put into the GLM PCA as covariates for adjustment before you do the dimension reduction. Okay, so the goal of linear dimension reduction is that we want to, sorry, I think I skipped this step as well, the highly variable gene. We want to use fewer dimensions than what we already have selected. So for highly variable genes, they are selected after the normalization, and basically we will reduce the total gene number down from, say, for human, we might start with 20,000 genes, and the filtering step to remove unexpressed genes may already remove some of them. And then we might end up like saying having 8,000, 7,000 genes. But that number will be further reduced down to the default is 2,000 genes in Seurat. So again, I think I mentioned that the 2,000 is an arbitrary number. It should depend on the data set. But what we hope to do this highly variable gene selection step is that we believe this is another assumption. That is, only the genes with much larger variance given the mean. The mean is basically the gene's mean expression. We think because for count data, mean and variance are related, like I said, for Poisson, for negative binomial. So that's what I want to say. If the gene has a large variance relative to the mean, then we think the gene's variance is likely biologically informative. So we want to keep the gene. But please note that if we use the normalized data, and if we can confidently say that the variance is already unrelated to the mean, then in this step, you just need to use the variance to select highly variable genes because the mean effect has been removed. However, if you use unnormalized data as count data, then when you say highly variable gene, you must account for the mean, since the mean is related to the variance. So therefore, I think another thing I said, which is quite interesting, is that we noticed that in one option for highly variable gene selection in Seurat, you used unnormalized data instead of normalized data. So that's another thing we need to be careful about. So once you normalize data, which steps further you want to downstream, you want to use normalized data instead of unnormalized data. That's another thing that can be tricky. All right, so coming back to the linear dimension reduction. So usually we use PC instead of highly variable genes after PCA. So the number of PCs is another parameter you need to decide. It can vary from 10 to 50. So it's a number in that order, but much smaller than 2,000. So what we hope to do in this step is to avoid the so-called curse of dimensionality. The curse of dimensionality says that if the dimension of your data goes up, if data becomes high dimensional, then you would have just curves. That is, Euclidean distances between data points will become not so informative. Okay, so Euclidean distance meaning will not be interpretable informative in the sense that you may have data points far away in Euclidean distance, even though they should be similar. So in that case, people want to work with a much smaller dimension if the smaller dimension can well preserve most of the variances in the high dimension. So that's why PCA is a suitable technique, because it is designed to preserve the variance at a much lower dimension. All right, so with that, we would use PCs. And GLM PCA also provides PCs. And on a related note, I talked about NMF because it is also linear. Okay, so even though NMF is not used in single cell data analysis for most of the papers in SURAT, but it is a very useful technique to know. If you remember that Nature paper we talk about in NMF, it is shown that by using a non-negative decomposition, factorization of your input matrix, which is also non-negative, you can better have a sparse representation. So every new dimension is only a sparse combination of the original features. And also because of negativity, non-negativity doesn't allow you to have positive and negative contributions from features that may cancel each other out, then they will force the features to have either zero contribution or positive contribution. So that will encourage sparsity. And the sparsity will lead to better interpretability. So you will say, oh, this new feature, just like my feature one, is explained by these original features. That will give you some explanation. So that's NMF. And then we talk about PNMF, projective non-negative matrix factorization, which is in the middle between PCA and NMF. So it satisfies this projective property of PCA. So with this projection matrix or loading matrix, you can project new data. A new data set which you didn't apply the dimension reduction, but you can use your first data set's loading matrix on the second data set to project that data set to the same space. That's advantageous. That's what PCA can do. That's what PNMF can do, but NMF does not. Okay, it also carries the non-negativity of NMF. So that's why you share the sparsity interpretability as NMF. And in my group, we have applied this PNMF to single cell data, and we found that it can be a very useful way to select the informative genes, which can separate cell types, even though you don't know the cell types beforehand. But SCPNMF can help you select the so-called distinguishing genes. Okay, so also we have talked about ICA, independent component analysis. So it's different from PCA in the assumption that it wants the signals, it wants to basically make it identify new features, be independent sources or independent signals. So ICA is most commonly used for signal processing, such as decomposing mixed audio signals into independent audio sources. And this independence assumption is different from the orthogonal assumption used in PCA. The difference is what? The difference is that for PCA, the orthogonality is what of the PCs is what you observe. You can use your data to check that indeed the PCs are orthogonal. So basically they have zero correlation and they are perpendicular in the space, in the PC space. But what we mean by independence of the ICs in ICA, that is based on distributional assumption. So the independence is what we use to solve the problem using a maximum likelihood estimation approach. So I don't know how many of you have learned about maximum likelihood estimation because this is not we cover in this class. But it is very useful, or I would say it is like a cornerstone of statistics. Basically, if you have a model, you have parameters to estimate, then maximum likelihood estimation is the most standard approach if you can write down the likelihood of your model given your data. So therefore, okay, so ICA would rely on that level of distribution assumption to be solved. And we show that, and we want to clarify that ICA is most suitable for non-Gaussian data. Instead of Gaussian data, the intuition underlying is that for Gaussian data, you can rotate it as you rotate, as you rotate your data points in a high dimensional space by changing the directions or rotate the whole coordinate system. You can rotate the whole coordinate system and still keep the data as Gaussian. And so therefore, when you do the decomposition, it's not so clear which direction you want to decompose the signals as independent. There are so many ways to do that because ICA doesn't have this maximum variance principle underlying. So you cannot do the same thing as PCA. But that rotation property will make ICA without unique solutions if applied to Gaussian data. But for non-Gaussian data, we don't have this problem. So it's possible to have a unique solution to decompose the signals into independent signals. And we have shown that how you can decompose the mixed image, right? Which is mixed from two pictures into two pictures using ICA. Okay, so linear dimension reduction to summarize PCA, GLM PCA, NMF, PNMF, and ICA. Okay, so regarding the determination of the dimensionality of or the number of PCs, we talked about this elbow plot approach, and we also talk about the needle method, which can automatically detect the knee, right, in that elbow plot. It's a kind of like a data-driven way for choosing the number of PCs. Another way is that, oh, I have a threshold in mind. I want the PCs I keep to retain at least 80% of my total variance. If that's what you want, you can choose number PC. But the problem with that is for single cell data, usually if we only keep say 10 PCs or 20 PCs, the proportional variance we can preserve may be far below 80%. I think this 80% or 90% or 20% is actually depending on what you believe the signal to noise ratio is in your data. Because if the signal to noise ratio is too low, then you have so much noise that if you want to preserve a large proportion of your variance, it doesn't make sense. Because what you preserve might be the variance from noise instead of signal. So how to determine the signal to noise ratio in your data is another challenge. Anyway, so I'm posing the open questions for you to keep thinking. Okay, so then with the PCs, clustering. So for clustering, we talk about several classical methods and then what's being used in SURAT. Classical methods include k-means and hierarchical clustering. K-means is the most standard one and widely used one that requires you to specify K beforehand. I need to say what is the number of clusters I want to find. Hierarchical clustering doesn't require you to specify K beforehand. So we talk about this agglomerative clustering algorithm, which means that I start from n cells and I gradually combine them into bigger and bigger clusters and build a hierarchy of cells. So given the hierarchy, we can decide where to cut. And for the cell hierarchy build, we need a distance metric. We also need a distance metric for k-means clustering, but by default, we use Euclidean distance in the PC space. For hierarchical cluster, we need to define the distance between two cells, and we also need to define the distance between two clusters, between two already built clusters. So we talk about single linkage, complete linkage, and average linkage as ways to establish cluster distances. Okay, and with hierarchical clustering, the advantage is that you will build a hierarchy and then you can decide where to cut. And the different layers or different resolution of your clusters will be nested. So that nested nature is something you don't have for single cell, for k-means clustering. For different k, you just have to run k-means from the beginning. You cannot use the previous k-means results with a bigger k or smaller k. Okay, then besides these two classical algorithms that operate on distances, that operate on Euclidean distances, we have graph-based clustering. And the graph-based clustering is closely related to the concept called manifold learning. We believe that the data points are not in just a linear or like a bow in the space, but basically we have to uncover some interesting low-dimensional manifold structure in the PC space. So if data are in a manifold like a Swiss roll, we know that Euclidean distances may not be informative. So therefore, we use a graph to approximate the manifold. And the idea is that we will define neighborhoods, right? Either by KNN or by epsilon neighborhoods. So then we can connect every dot to its neighbors only. And then we hope this neighborhood graph preserves the manifold or approximates the manifold. And we talk about two ways to build a graph, which are both in Surat. One is KNN, connecting every cell to its k nearest neighbors in PC space based on Euclidean distance, or SNN, shared nearest neighbor graph. By shared nearest neighbor graph, we look at two cells, 2 points, and we check their KNNs. Two sets of KNNs, we look at the intersection of the two sets and the union of the two sets. And the intersection size over the union size is what we call the Jacob index. We use that to indicate the similarity between two cells and put that as the edge rate. And in Surat, when they build this SNN graph, I think there is a minimum threshold on the Jacob index. Below that, you don't keep the edge. Okay, so with the neighborhood graph, then if we want to define cell clusters, we apply a graph-based clustering algorithm. And specifically, we talk about two heuristic algorithms used in Surat. One is called Louvain, one is called Leiden. So Leiden is a refined algorithm based on Louvain to ensure that the points in each cluster remain connected. That's the major change from Louvain to Leiden. But Louvain is implemented faster, so that's why Louvain is the default. Even though the Leiden paper claimed that Leiden should be faster, but this difference in computational speed between the paper and the Surat implementation is probably due to software implementation. So we also leave that as a point, as an open question. So the ways Louvain is doing is to maximize this property, or I would say quantity, called modularity. Modularity measures how tightly points are connecting one cluster and how little connected they are between clusters. So what they want to say, they want to encourage more within cluster connections than between cluster connections. Such a network will have a large modularity. Okay, so Louvain will do that in a heuristic way by changing the cluster labels of data points to greedily, I would say, maximize the modularity, but there's no guarantee for global optimization. And also, the order you go through your nodes or data points in Louvain might change the result. Okay, but the good thing is that Louvain is very fast. So that's why for large graph, you cannot possibly do something like spectral clustering, which is also applied to a graph. It might be too slow, but Louvain can give you the clusters, cell clusters. Okay, once we have the cell clusters, then we do nonlinear dimension reduction for just 2D, two-dimensional visualization. And we talk about UMAP t-SNE, where these are what's used in Surat. But much earlier in 2000, in the science paper, there were two methods introduced. One is called ISOMAP, one is called LLE. For ISOMAP, the idea is to apply the MDS, which we also talk about in linear dimension reduction to preserve distances in 2D. Applies MDS not to Euclidean distances in the high dimensional space, but to geodesic distance, which is inferred from the graph. So basically, the geodesic distance is closely connected to this graph building we talked about earlier regarding manifold learning. To preserve the geodesic distance, that's isomap. And LLE is try to find low dimensional embeddings by assuming that there's a linear relationship so that you can predict every point based on its neighboring points using a linear regression. And you keep the linear regression weights you learn from high dimensional space, you keep the same weights in low dimensional space, and then use that to find the low dimensional embeddings. That's LLE. Okay, then UMAP t-SNE stem from another idea from SNE. SNE is the earlier one that is the ancestor of t-SNE, and then UMAP modifies them. So the idea is that we define some kind of density in the high dimensional space for each data point I using all the neighboring points, and we use a Gaussian kernel to basically transform their Euclidean distances into densities. And we scale the densities to become probabilities. And we hope to preserve the same probabilities in low dimensional space, in 2D space. So the idea is that we want to look for the low dimensional embeddings, 2D embeddings, by preserving those probabilities. And there are two major changes from SNE to t-SNE. The low dimensional density in t-SNE is based on a T distribution instead of a two-dimensional Gaussian distribution. And also, in the probability calculation, t-SNE made the probability from I to J and J to I symmetric, while in SNE, they're not symmetric. And for both SNE and t-SNE, there is an important parameter called the perplexity. The perplexity is how you define the neighborhood size when you define the Gaussian kernel. So if you have a larger perplexity, your data points will be more connected with fewer clusters, but a smaller perplexity will give you very fine clusters. And UMAP, the major change is the objective function or loss function. So in SNE and t-SNE, we use the KL divergence. But KL divergence does not preserve the probability well if the high dimensional probability is close to 0. But UMAP uses the cross entropy as the loss function, which avoids that issue. So you have better chance to preserve both the high dimensional P probability that's closer to 1 or closer to 0. So you have better preservability of that. Okay, but UMAP has two parameters. One is the number of neighbors, and the other is the minimum distance. And both parameters will change the visualization. So it is claimed that UMAP has better preservation of the global topology of cells than t-SNE by not having too many small clusters. But I think there is another paper, which I didn't mention in this class, that says, oh, the advantage of UMAP is mostly about the initialization, because they in the UMAP paper that claimed this advantage, a different initialization is used for UMAP than t-SNE. So like UMAP initializes from PCA and t-SNE initializes from random initialization. That will make a difference. So therefore, I would say some people use UMAP because I think in the current implementation, it is also faster than t-SNE. So it has the computational advantage. But anyway, for both methods, we should use them cautiously, instead, especially if we want to draw some conclusions about the positions of clusters in the 2D visualization. We have to be cautious. We have to be aware that they are not designed to preserve the global structure of your data. So my group developed a method called SED, which is hopefully trying to let you see which cells embeddings might be dubious relative to other cells. So then you should be cautious about interpreting the dubious embeddings and also optimize the hyperparameters for t-SNE and UMAP. Okay, finally, what we have so far is that we have obtained the cell clusters and we have seen them 2D. And then the last step is to find some marker genes for the cell clusters, then we can annotate the cell clusters as some cell types. This is what you will do in your final project. And for this marker gene part, we talk about differential expression, which is widely used in bioinformatics. How do you find differential expressed genes or differential expressed features between two samples or here between cell clusters? So for this one, what I focused on in our last one lecture is about what is a hypothesis test in the frequentist sense, right? We talk about the null hypothesis, alternative hypothesis, test statistic construction, null distribution of the test statistic under the null hypothesis, p-value calculation, and some common mistakes you might encounter. So then once we get a p-value per gene, and also we talk about the two-sample t-test, Wilcoxon rank sum test as the standard approach, classical approach, and Wilcoxon rank sum test is the default used in Surat. So once we have one p-value per gene, we need to consider multiple testing correction. Okay, for multiple testing correction is to avoid the inflated type 1 errors as the number of tests goes up. We have talked about the family-wise error rate and its related Bonferroni correction, which is conservative. And then we talk about a new criterion, which is less conservative, which is the false discovery rate, and has become very popular when the number of tests is large. And for FDR control, we talk about the Benjamini-Hochberg correction, which is based on ordered p-values and increasing thresholds. And we also talked about the p-value free FDR control using more recent approaches. So once we have the differential expressed features, we can annotate cell types. But one thing I noticed in the last note is the double dipping issue, which is that you use the same data to do clustering and DE analysis, then naturally you will find a lot of DE genes. And my group has developed an approach to circumvent or avoid this double dipping issue. So that's all. So basically, we talk about several major statistical concepts used in bioinformatics in the context of Surat. And I want to emphasize once again that many methods are not just for single cell. They can be used in many settings, such as normalization, such as linear dimension reduction, such as clustering, nonlinear dimension reduction, and differential expression analysis. Okay, so that's all I want to say about the course material review. If you have any questions about these contents, you can ask for the remaining time. But I want to give the floor to you so you will have time to answer any questions you might have for this class and for the final project.